Okay. Welcome everybody. Today we are continuing the Atheronet Cardioscope webinar series. As you know, all these webinars are being recorded and later on are also available on our two YouTube channels, on Cardioscope YouTube channel and on Atheronet YouTube channel. Today we have a very interesting lecture by Angelika Chroni from Greece, that is focused on high throughput methods for evaluation of changes in composition of and functions of HDL, as well as for the discovery of compounds that improve HDL functionality. We are digressing a little bit there, diverging from the RNA world that we were so much obsessed and focused on in the previous couple of lectures. A few words about the Dr. Chroni. She's a research director at the Institute of Bioscience and Application National Center for Scientific Research, Democritus. She holds a PhD in biochemistry. She worked at Boston University Medical School from 2000 to 2005 as a postdoc researcher. Her research is mostly focused on pathogenesis of atherosclerosis and Alzheimer's disease, and the ultimate goal of her work is to elucidate the molecular mechanisms that are involved in the development of these diseases. She also received in 2009 L'Oreal UNESCO Award for Women in Science. So without further ado, Angeliki, you can start, and then you have around 45 to 50 minutes to kind of go through the lecture, and then we have 10 to 15 minutes to discuss and to ask questions. Thank you.
Okay, thank you. Good afternoon. Thank you very much for the invitation. Today I'm going to talk about high throughput methods for evaluation of changes in composition and functions of HDL. My presentation has three parts. The first part is about, I will briefly talk about the associations between HDL levels, composition, function, and atherosclerotic cardiovascular disease. Then I will talk about the high throughput methods that have been developed for the evaluation of changes in HDL composition and function. And finally, I will present briefly some recent data on the use of such methods for the discovery of compounds that improve HDL functionality. Okay. You know, you are aware about the relationship between LDL cholesterol and cardiovascular disease. It is well known that cardiovascular disease is characterized by high levels of LDL cholesterol, and these high levels have a causal role in the development and progression of atherosclerotic cardiovascular disease. Therefore, the measurement of LDL cholesterol levels play a central role in risk assessment for cardiovascular disease in all international guidelines, and various medications that lower these levels have been developed, such as statins, ezetimibe, PCSK9 inhibitors, bempedoic acid. But what is the relationship between HDL cholesterol and cardiovascular disease? Several large-scale epidemiological studies have shown a consistent and long-standing observation of increased cardiovascular events in participants with low HDL cholesterol. However, the inverse association of plasma HDL cholesterol levels with cardiovascular disease risk has not been translated into positive outcomes in the clinical trials of drugs that were developed so that to raise HDL cholesterol levels. Furthermore, Mendelian randomization studies found no evidence of a causal relationship between HDL cholesterol levels and cardiovascular risk. The failure of the clinical trials and the results of Mendelian randomization analysis appear to be related to the fact that HDL cholesterol levels do not reflect the function of HDL. So why are we still interested in HDL as a diagnostic and therapeutic target? The first reason is that because even after the introduction of the most effective LDL cholesterol lowering therapies, there is still residual risk for atherosclerotic cardiovascular disease. The other reasons are that the low HDL cholesterol levels are consistently found in conditions of atherogenic dyslipidemia, and the main reason is that HDL do possess arteroprotective functions. HDL is a complex family of different particles that composed of subspecies that differ in size, density, shape, charge, and composition. And these particles undergo continuous remodeling in the circulation. So most of HDL particles are spherical, but can be found also in discoidal forms. There is a diversity in the density, in diameter, density, electrophoretic mobility, also in their composition. The main apolipoprotein protein is A1, but also there are particles that contain ApoA2, ApoE can be also found in HDL, and proteomic analysis showed the presence of more than 300 proteins, proteins that are involved in lipid transport and lipoprotein integrity as expected. Also, lipolytic hydrolytic enzymes are found in HDL, and other proteins such as members of complement pathway, protease inhibitors, acute phase response proteins, and others. Furthermore, more than 300 lipid species are found in HDL, as well as hydrophobic metabolites and non-coding RNAs, including microRNAs. And this compositional complexity leads also to functional complexity. And HDL has several functions, mostly atheroprotective, that include cholesterol efflux capacity, antioxidant, anti-inflammatory, immune regulating functions. Now, under various pathogenic situations like chronic diseases such as coronary heart disease, rheumatic and autoimmune diseases, and other diseases, HDL can lose its protective functions and even gain adverse functions. So under these conditions, HDL and ApoA1 can undergo oxidative modification or truncation. The peroxonase 1, which is an enzyme with antioxidant activities, also can undergo oxidative modifications. There is an increase in HDL of molecules with pro-oxidant and pro-inflammatory functions such as serum amyloid A, and other modifications happen that impair the atheroprotective properties of HDL. Now, the most three more important and more studied functions of atheroprotective functions of HDL are its capacity to promote cholesterol efflux, the antioxidant properties, and anti-inflammatory properties. Cholesterol efflux capacity is the most examined atheroprotective function of HDL, which is a major part of the reverse cholesterol transport pathway. So HDL can remove cholesterol from the macrophage foam cells that are found in plaques and deliver it through a pathway to the liver for processing and exertion into bile. Cholesterol efflux is mediated by cholesterol transporter ABCA1 that interacts mostly with lipid-poor ApoA1, as well as with cholesterol transporter ABCG1 and HDL receptor SRB1. Both ABCG1 and SRB1 interact with HDL particles, lipoprotein particles, and promote the uptake of cholesterol by HDL, the efflux of cholesterol by HDL. Important function is the anti-inflammatory function of HDL because inflammation is known that plays a key role in the pathogenesis of atherosclerosis. HDL reduces inflammation in various cell types that mostly include endothelial cells and macrophages. I won't provide many details on these pathways, but just briefly, HDL can downregulate the cell adhesion molecules VCAM1 and ICAM1, can prevent the expression of IL-8 and monocyte chemoattractant protein, and can upregulate endothelial NO induction and availability, while in macrophages can regulate mostly the production of cytokines, including TNF-alpha and other interleukin-1 beta and other cytokines. And regarding its antioxidant function, HDL and proteins that are found in HDL, such as ApoA1, pyroxonase 1, PAF-acetyl hydrolase, and LCAT, can prevent cellular oxidative stress and LDL modifications by inhibiting either the formation of lipid hydroperoxides or hydrolyzing oxidized phospholipids. So these various HDL functions have been associated with cardiovascular events and mortality. Also, several HDL components that affect the functions of HDL are associated with the presence of acute or chronic cardiovascular disease and also react to therapeutic interventions. And therefore, HDL components and functions could serve as biomarkers to improve the identification of individuals that are at risk for cardiovascular disease, treatment stratification, and monitoring of these individuals. Several studies have examined the association of cholesterol efflux capacity with cardiovascular events, and the majority of these studies have shown an inverse correlation between cholesterol efflux capacity and cardiovascular events. In a meta-analysis of that kind of studies that was published in 2020, it was found that cholesterol efflux capacity, greater cholesterol efflux capacity of HDL was associated with lower risk of cardiovascular disease. This meta-analysis included 8 prospective studies with more than 11,000 participants. And also a subset of these studies showed that lower mortality was related to higher cholesterol efflux capacity of HDL. In the same paper, it was examining the association of HDL antioxidant capacity with major adverse cardiovascular events and all-cause mortality. Only 2 studies examining the association of antioxidant capacity of HDL with cardiovascular disease, and there was no statistical significant association between these 2 parameters, but it was reported that the higher HDL antioxidant capacity correlated to lower all-cause mortality risk. Also, the relationship of cardiovascular events with HDL anti-inflammatory function has been also examined. It was examining the ability of HDL to suppress the TNF-alpha-induced VCAM mRNA expression in endothelial cells. And it was found that impaired HDL anti-inflammatory ability was associated with new events in patients with acute myocardial infarction and with incident cardiovascular events in a general population cohort. The risk of anti-inflammatory function with cardiovascular events showed an inverse correlation as compared to the association of HDL cholesterol levels with cardiovascular disease that showed no correlation. Also, some other parameters of HDL has shown correlation with the risk of cardiovascular disease. HDL particle number that determines cholesterol efflux capacity is inversely associated with the risk for cardiovascular disease. Serum amyloid A that inhibits cholesterol efflux has a positive association with cardiovascular disease. Pyroxenase 1 shows an inverse association. Plasmalogens, specific plasmalogens that exert antioxidative functions also show an inverse correlation with cardiovascular disease, as well as sphingosine 1-phosphate. So it appears that HDL functions and composition are associated with cardiovascular disease and could serve as biomarkers for risk assessment and treatment monitoring, but currently remain recent research tools. Detailed analysis of HDL components and functions by methods that combine both high resolution and high throughput consist the main technical challenge in this field. So in the next part of my presentation, I'm going to present the high throughput methods that have been developed for evaluation of changes in composition and functions of HDL. In order to study HDL function and composition, it is necessary as a first step to isolate HDL from the rest of lipoproteins in plasma or serum. Several methods can be used. The method that has been mostly used for HDL isolation is the two-step gradient density ultracentrifugation, that involves 2 overnight ultracentrifugation steps. In the first step, HDL is isolated from VLDL, LDL, and in the second step, HDL is isolated from the rest of HDL particles are isolated from the rest of proteins that are found in plasma or serum. This method is time intensive and low throughput. It's not appropriate for automation. So other methods have been developed. These methods are based on the precipitation of ApoB lipoproteins from plasma or serum. And for this precipitation, they use either a synthetic polymeric compound, which is polyethylene glycol 6000, or polyions like dextran sulfate 50,000, along with magnesium cation. So HDL is mixed by any of these compounds, then a centrifugation step follows, and HDL is isolated in the supernatant. The advantages of these methods are the cost, are cheap methods. They have clinical applicability and can be brought to high throughput. A limitation is that other proteins can be found in HDL supernatant. However, comparison of functional properties of HDL that is isolated by this method as compared to HDL isolated by gradient density ultracentrifugation showed similar functional properties such as cholesterol efflux capacity, inhibition of LDL oxidation, and anti-inflammatory properties of HDL. As I said in the previous slide, cholesterol efflux capacity has been associated in many studies with the assessment of cardiovascular risk. And many assays have been developed in order to have a high throughput method that would be representative of cholesterol efflux capacity of HDL. The gold standard to measure this capacity is a cell-based assay that in this assay we use J774 macrophages, but also other type of macrophages like RAW or THP macrophages, which are labeled with radioactively tagged cholesterol or fluorescently tagged cholesterol. Then the labeling proceeds for 24 hours, and then the next day the cells usually are treated with a cyclic MP analog that upregulates the expression of ABCA1. And then the other day, HDL particles, usually ApoB depleted serum, is added in cell medium, and following an incubation, usually 4 to 6 hours, we separate cell medium from cells that are lysed, and we measure their radioactivity in the medium and the lysate, or fluorescence intensity if we have used fluorescently labeled cholesterol, and we can calculate the cholesterol efflux capacity of HDL. By using this method, we can determine total cholesterol efflux capacity, which represents cholesterol efflux mediated by ABCA1, ABCG1, and SRB1 transporters and receptor. It is clear that this assay is a time-consuming assay and labor-intensive. It requires culture cells, it's low throughput, and it is an assay that is hard to transform into a clinical diagnostic assay. So many efforts have been made in order a cell-free assay to be developed that could replace the cell-based assay. One type of assays measures the exchange of ApoA1, actually the binding of lipid-free ApoA1 to HDL. In one type of that kind of assay, spin-labeled lipid-free ApoA1 was incubated with ApoB-depleted plasma or serum, and then and then following this incubation, ApoA1 is incorporated into HDL, and quantitation follows by electron paramagnetic resonance spectrometry. This assay associates very well with a cell-based assay. It is high throughput, but requires specialized instrumentation. So a variation of this assay was developed it used a dual fluorescent labeled ApoA1. ApoA1 was labeled with NBD and Alexa 647 probes, and it was again incubated with HDL. And the fluorescence, both of MBD and Alexa 647, was read over time. And upon the exchange of ApoA1 into HDL, the MBD to Alexa 647 emission ratio increases over time. NBD is a probe and that its fluorescence changes following the lipidation of ApoA1 when it binds to HDL, while Alexa 647 is lipidation insensitive. This assay shows a statistical significant correlation with a cell-based assay, and an even better correlation with the ABCA1 independent cholesterol efflux, which is cholesterol efflux dependent to ABCG1 and SRB1. It's a high throughput assay, and in a small clinical study, it was shown that it was shown that a low ApoA1 exchange rate was associated with increased incident risk for major adverse cardiovascular events. Another assay, cell-free assay that was developed in order to replace the cell-based cholesterol efflux capacity assay involves the uptake of cholesterol by HDL. HDL is incubated with body-labeled cholesterol. Then HDL is captured by a specific antibody against ApoA1, and then the fluorescence intensity of labeled cholesterol, which is incorporated into HDL, is measured. Again, this assay, which is based on the uptake of cholesterol by HDL, shows a nice correlation with a cell-based cholesterol efflux capacity assay. And a variation of this assay has also been developed. Body P cholesterol was substituted by biotin PEG-labeled cholesterol. And following incubation, HDL is captured by an ApoA1 antibody that is bound into magnetic particles. And this step helps the automation of the assay. And then alkaline phosphatase, which is conjugated into streptavidin, is added. And following the addition of specific substrate, chemiluminescence, the chemiluminescence signal is measured. This cholesterol uptake capacity of HDL was found to be inversely associated with the requirement for revascularization in a small clinical study. Another type of assay involves the exchange of cholesterol between artificial liposomes and HDL. So in this assay, liposomes made by PC cholesterol and fluorescently labeled cholesterol were immobilized in cefacryl gel beads. These beads were incubated with HDL. Centrifugation followed to separate the beads from the supernatant that contains HDL with the fluorescently labeled cholesterol that has been moved from the artificial liposomes to HDL, and the fluorescence is measurement. This cholesterol exchange assay seems to correlate very well with a cell-based assay. And the same group, a couple of years later, developed a variety of this assay. Gel beads were replaced with magnetic beads to facilitate the automation of assay by skipping the centrifugation process. And it seems that the two assays using either the gel beads or the magnetic beads correlate very well. Lastly, another cell-free assay that could replace the cell-based assay has been developed by Alan Ramley's lab. And it is an assay that measures the phospholipid efflux of HDL. The idea is that during the lipid efflux process from cells, not only cholesterol, but phospholipids are also removed. So artificial lipid donor particles were developed that were calcium silicate hydrate crystals coated with PC cholesterol and labeled with fluorescence tagged phosphatidyl ethanolamine. And in this assay, these crystals are incubated with whole plasma or serum, not isolated HDL, whole plasma. And it appears that following incubation of crystals with lipoproteins, cholesterol can be found both in LDL, VLDL, and HDL, but the label phosphatidyl ethanolamine is found only in HDL. So by measuring fluorescence in the medium, so following this incubation, a centrifugation step must be applied to get rid of the crystals from to get rid of the crystals. So measuring the fluorescence intensity in this supernatant, we can quantitate the HDL phospholipid efflux capacity. And actually, it was proposed that in this process of transfer of PE from the artificial particles to HDL, ApoA1 has an important role, and the whole process mimics the ABCA1 modified cell membrane lipid domains on cells by permitting ApoA1 to insert into the artificial particles and then solubilize and remove phospholipids. It seems that this HDL phospholipid efflux correlates quite well with a cell-based cholesterol efflux assay, and use of this assay in two small clinical studies showed that HDL-specific phospholipid efflux is inversely associated with both with prevalent and incident coronary artery disease. So these were the assays, the cell-free assays that are being developed to replace the cell-based cholesterol efflux capacity assay. For evaluation HDL antioxidant capacity, two cell-free assays are used. The first one is the T-bars assay, in which LDL is oxidized by a specific compound in the presence or absence of HDL, and then the accumulation of thiobarbituric acid reactive substances as a measure of oxidative modification is measured by fluorescence spectroscopy. So in the presence of HDL, T-bars are expected to be lower as compared to LDL that has been oxidized in the absence of HDL. Another assay more extensively used is the DCF assay, in which LDL is mildly oxidized by exposure to air. And then this mildly oxidized LDL is incubated in the absence and presence of HDL, along with a non-fluorescent probe, which is called DCFH2. And this probe, upon interaction with oxidants, is oxidized to another form, the DCF, which emits fluorescence. So by measurement the fluorescent intensity, we get an estimate of the capacity of HDL to suppress the oxidative potential of oxidized LDL, and low fluorescence signal indicates increased antioxidant activity of HDL. Both assays are high throughput. And this DCF assay was developed almost 25 years ago by Fogelman's and Navab group. And as we can see here, normal HDL results in low fluorescence signal, while HDL from patients with coronary arteriosclerosis result in high DCF signal. when incubated with oxidized LDL, actually the signal is higher than the signal that we get only by oxidized LDL. And it was when this assay was developed, it was, excuse me, it was examined, it was compared with a cell-based assay that measured the antioxidant capacity of HDL. In this cell-based assay, oxidized LDL was added to co-cultures of human aortic endothelial cells and human aortic smooth muscle cells in the presence of HDL from patients or from controls. And following the incubation, the supernatants from the cell co-cultures were tested for their monocyte chemotactic activity. And as we can see here, normal HDL results in low migration of monocytes, while HDL from patients with coronary atherosclerosis results in high migration of monocytes. And there was a very nice correlation between the two assays, the cell-free and the cell-based assay. Actually, the results are virtually identical between the two assays. Also, some other assays can be used for quantitation of other parameters related to HDL and related to the functions of HDL. So the number of HDL particles can be measured by NMR, and it has been shown in a pool cohort of 4 large population studies that HDL particle concentration was inversely associated with myocardial infarction and ischemic stroke, and Mendelian randomization studies showed that HDL characteristics that were measured by NMR had a significant causal association with coronary heart disease risk. So NMR spectroscopy is high throughput and can provide useful information, but this is a technique that requires specialized instrumentation. Similarly, lipidomic analysis can be useful. Specific lysophospholipid species of HDL were found capable to discriminate patients presenting acute coronary syndrome from those with stable coronary artery disease. And HDL plasmalogens that scavenge oxygen radicals have shown inverse association with both stable and acute coronary artery disease. But again, we need specialized instruments for this analysis. Much simpler are the measurements of levels of proteins or enzymes, or the activity of enzymes associated to HDL particles that affect the functional properties of HDL. So the levels of serum amyloid A1 that impair antioxidant and the anti-inflammatory properties of HDL can be measured by ELISA. The levels or activity of myeloperoxidase can be again easily measured by the levels by ELISA, the activity by simple enzymic reactions. The enzyme can react with hydrogen peroxide, which oxidizes specific substrates to either colored or fluorescent products, or by a combination of both ELISA and activity assay. These are rapid, high throughput, and highly reproducible assays, but the disadvantage is that there is a lack of evidence in a large clinical trial to predict coronary artery disease events. The activity of pyroxenase 1, an enzyme that displays antioxidant capacity, it is proposed to oxidize to hydrolysis oxidized phospholipids, can be easily measured. So PON1 is associated in circulation mostly with HDL particles. PON1 activity has been found to be inversely associated with cardiovascular disease incidence in humans. In animals, knockout of PON1 and overexpression lead to increased and decreased atherosclerosis respectively. And as I said, the hydrolysis of oxidized phospholipids represents the main function of this enzyme. So by simple kinetic absorbance assays, we can measure either the paraoxonase activity of the enzyme by using a substrate paraoxon, which is considered the most specific substrate for this enzyme, or we can measure the aryl esterase activity of PON1 by using phenylacetate as a substrate. And this activity is one of the best, this substrate is one of the best substrates of this enzyme, and the activity, aryl esterase activity is much stronger activity than the pyroxenase activity. And the absorbance or hydrolysis products in these assays is recorded over time. It is a very rapid, high throughput, and reproducible assay. And before closing this presentation, I would like to present very briefly, some recent data that was based on such a high throughput method than those that I presented, and was used for with the aim to discover compounds that improve HDL functionality. So agents that modulate HDL functionality have not been discovered, and we wanted to see whether we could identify such molecules. With screened a library of marketed drugs, almost 1,000 compounds, which are drugs approved by FDA, in an effort to identify molecules that can increase human HDL-associated PON1 activity. So we'll skip this slide because I talked about PON1 before. We, as an enzyme source, we used pooled human HDL, and we examined each compound we examined the compounds for their capacity to increase the paraoxonase activity of PON1 that is associated on HDL. The first screening resulted in about 10 potential activators. This then further repeat was performed, and further validation of selected compounds. And we also measured the capacity of these compounds to increase the aryl esterase activity of the enzyme. And we found 7 compounds that can induce a more than 5% increase of pyroxenase activity, and 4 of them increased aryl esterase activity. And the strongest activation was observed by a drug which is called dantrolene. Since pyroxenase 1 in humans has 2 very common polymorphisms, the L55M and Q192R, which have been proposed to affect enzyme activity and cardiovascular risk. We wanted to examine the effect of the selected 7 compounds on the activity of these PON1 variants by using as enzyme source HDL from carriers of these variants. So we used carriers of wild type PON1, homozygotes for the L55M variant, heterozygotes for this variant, and heterozygotes for the Q192R. And we found that dantrolene promoted the activation of all variant forms of PON1, and actually the most strong activation was observed for L55M variant. This is the structure of dantrolene. It's a postsynaptic muscle relaxant that has been approved by FDA for the treatment of malignant hyperthermia. And in order to confirm that dantrolene has the capacity to activate PON1, we expressed wild type and the L55M PON1 in recombinant form, and examining the capacity of dantrolene at increasing concentrations to induce the activation of PON1. It was confirmed that dantrolene is a PON1 activator, and actually the activation was more pronounced for the variant L55M PON1. And further enzymatic analysis showed that dantrolene induces the Vmax of the enzymatic reaction for both wild type and variant PON1. Also, we performed molecular docking of dantrolene onto PON1 structure. This analysis showed extensive interactions of dantrolene with the enzyme. Dantrolene can make direct or indirect interactions with the enzyme's active site, but it does not block the substrate approach. This observation is consistent with the ability of dantrolene to activate PON1. And also, it was shown that a specific part of dantrolene can potentially coordinate a calcium atom. I forgot to say that PON1 is a calcium-dependent enzyme. By specific part of its structure, dantrolene can have an important role in PON1 activation. And finally, the last question that we asked was whether dantrolene can activate PON1 in vivo. So we performed in vivo studies in collaboration with the University of Crete and with the laboratory of Professor Prodromos Sidiropoulos and Professor Dimitris Kardassis. And for these studies, we used a mouse model of rheumatoid arthritis. We used this model because it is well known that the activity of PON1 is decreased, and the HDL functionality, including antioxidative function, is impaired in patients with rheumatoid arthritis. And since it is well known that in such chronic autoimmune diseases, HDL function is affected, and there is an association of HDL dysfunction with increased risk for cardiovascular disease in these patients. So we investigated whether the administration of dantrolene in an arthritic mouse model can increase PON1 activity and improve the HDL dysfunction that is present due to the inflammatory environment that is present in these animals. So for this, for this animal model, arthritis is induced by injection of bovine collagen in the mice, followed by a boost injection after 21 days. And rheumatoid arthritis starts to be evident in day 28. And two protocols were followed. One, which is the preventive, protocol, in which dantrolene was administered along with the induction of arthritis, and the mice were sacrificed at day 35, while in the therapeutic protocol, the administration of dantrolene started along with the onset of arthritis, when arthritis was clinically evident, and mice were sacrificed on day 42. For the assessment of arthritis severity, the front and hind limbs of mice were graded for the presence of redness and swelling of joints, and also paw thickness was measured. So a score for arthritis can be calculated along with the measurement of paw thickness. And following the sacrifice of mice, serum is collected, joints, spleen, lymphatic nodes, for a series of analyses. I won't present all of the analysis, but the main findings are that both in the preventive protocol and the therapeutic protocol, the administration of dantrolene reduced arthritis severity and suppressed T cell responses. And also, we measured pyroxonase and aryl esterase activity and HDL antioxidant properties. And in both protocols, pyroxonase and aryl esterase activities are increased following the administration of dantrolene. And as we can see here, DCF fluorescence signal is much lower following administration of dantrolene in preventive protocol, and also lower in the therapeutic protocol, indicating that the administration of dantrolene before the onset of arthritis, as well as the therapeutic administration of this drug upon the onset of the first clinical symptoms, improved both pyroxonase 1 activity and HDL antioxidant capacity. So, using small molecules to enhance HDL-associated PON1 activity could be a tractable approach, leading possibly to novel therapeutics that target immune responses and atherosclerosis. This is a very recent study. It was published a couple of weeks ago. And just to finalize the overall talk, HDL atheroprotective functions have been associated with atherosclerotic cardiovascular disease. HDL components and functions may serve as biomarkers to improve identification, treatment stratification, and monitoring of individuals at risk for atherosclerotic cardiovascular disease. Several high-throughput cell-free assays have been developed in order to measure the functionality of HDL, and can be used for cardiovascular disease risk identification, therapy monitoring, and possibly for drug development. But the clinical application of these assays and the correlation with cardiovascular risk should be examined in well-designed case-control studies that are appropriately powered to provide a strong statistical significance that is clinically meaningful. Thank you very much for listening and for being here today. And I would like to receive any questions.
Thank you so much. This was quite an interesting overview of story behind HDL. As Dimitris usually say, this is the molecule, I mean, the particle that went ups and downs, many rises and pitfalls along the previous 20 years. And it's really nice to see that finally we now have the information why Mendelian randomization study where the results from these studies were a bit disappointing from HDL aficionados. But now it's really nice to see that we need to focus more on functionality of HDL than actually just on cholesterol concentration, which is just a small part of what HDL actually does. So thank you so much for this overview. I have maybe two questions to start the discussion, and then we can see if anybody else has any questions from the audience. If anybody has questions, you can use the chat, or you can raise your hand, and then we will unmute you, and then you can ask the questions. So for me, one question is regarding the first part of your story, where you explained overall principles of the mechanisms of reverse cholesterol transport and how HDL is actually evolving through this transport from small particles to large particles. Now, what I've have hard time to understand is actually there are two different opinions on this, and I always like to ask this question to the experts. So if we observe HDL3 particles to be predominant in the concentration wise in the blood of patients, does this mean it's a good thing or it's a bad thing? Because at some points, some schools of HDL will say, yeah, but it's of course good to have high HDL3 because that means that your capacity for reverse cholesterol transport is very big, and you can, a person can take so much of cholesterol because of that. But the other school of HDL is telling us that this is not a good thing, because when you have a lot of HDL3 particles, it means that reverse cholesterol transport does not work properly, and then as a consequence of this you have accumulation of HDL3. So is it really easy to give this kind of black and white binary code interpretation of this, or it needs to be context dependent?
Since it is not a matter of quantity but quality, I mean, we cannot just say, okay, we have a lot of this and it's good, or we have a lot of this and it's bad, but we need to know what is the composition of this HDL3 regarding proteins and lipids. It depends if is someone that's healthy or is someone that is has an autoimmune disease and the composition has been affected. However, the studies that have shown that very high HDL levels may be detrimental do exist, and this may be related with how HDL can mediate the uptake or the removal of cholesterol from the circulation.
Yeah. Can I comment on this, Mira?
Of course.
Yes.
Of course.
So yes, the new epidemiological data show that the association between HDL and CVD risk is U-shaped, right? So high levels, very low levels, and very high levels are not good. The risk is high. And of course, we know that very high levels are not good because all these inhibitors, CETP inhibitors, didn't work, which increased HDL levels. And also in mice, if you knock out SRP1, if you, mice, very high levels of HDL is not necessarily good for patients. So first of all, to have very high levels of HDL means something is wrong. Okay, the HDL cannot be cleared. Otherwise, you should not have so high levels of HDL. Maybe the receptor is not working, something is not working there. So the metabolism is disrupted. I think this is what is happening. The metabolism of HDL is disrupted, and this is not good. It's never good to disrupt the physiological metabolic pathway. It's better to enhance the existing pathway rather than disrupting the catabolic, let's say, pathway. So probably that's why these patients with very high levels of HDL are not protected, because the particles are not functional, something like that.
Okay, thank you. We have a question from Corinne. Let me just go down. Yeah, Corinne asks, is there an association between BMI and HDL functionality in CVD or healthy subjects?
BMI, I'm not, I don't have in my mind the exact study now, but I think that HDL is dysfunctional in cases of obesity.
Which causes diabetes. It's strongly associated with diabetes, and we know that diabetes causes dysfunctionality. So,
is it related to the changes in triglyceride metabolism and interaction between the LDL and HDL through the CTP and?
It could, and also I think there are lipidomic studies in subjects with obesity that have shown very significant changes in the lipidomic composition of HDL that could affect functions and the overall thyroprotective capacity of HDL.
Okay. Do we have more questions from the audience? I have a second question from my side, is related to the functional analysis. So we know that HDL has many functions, as you nicely mentioned and show in the graphs. But if we want to functionally analyze HDL, maybe it's not realistic to really assess all the different functions. I mean, in the clinical perspective, let's say we are discussing about translational potential. We want to go into clinics and having in the lab the test that measures certain aspects of HDL beside the concentration of cholesterol. Which function would you choose to start with? Which of those functions of HDL you would say has the biggest influence on overall cardiovascular risk? Would it be antioxidant potential, as you mentioned, through PON activity, or would it be something else?
I think cholesterol efflux capacity, which has extensively studied and has shown a very good correlation with cardiovascular disease risk, and that's why there are so many efforts to develop a cell-free assay that correlates very well with a cell-based cholesterol efflux capacity assay, which cannot be used in a high throughput setup.
Okay, thank you. Any more questions, Dimitri? Do you have something?
Yeah, maybe a comment. Yes, of course. All these assays that Angelica described could be used and could be useful in the clinic, but they do not replace cholesterol reflex capacity. I mean, this is for sure. I mean, this is something else. What we're measuring in this high throughput assays is not cholesterol reflex, it's binding of ApoA1 to lipids, etc. But the cholesterol reflex capacity cannot be easily measured in high throughput in the clinic. It's more, let's say, for research purposes, and there are many papers clinical papers using cholesterol reflex capacity to assess risk, etc., and they work very well. But to do routinely in the clinic, I think it's very difficult. You need special instruments, tissue cultures, radioactivity, perhaps fluorescence, etc. So I don't know if you, Angelica can comment on that, because the reflex capacity cannot be easily replaced in the clinic, and this is the real functionality of HDL.
Yes, but this is the idea, to replace this assay by finding an equivalent assay that associates very well with cholesterol reflex capacity assay and can replace it. But still, we need to do a large-scale clinical studies to see that indeed we can replace the cell-based assay, which is the gold standard.
So all these studies that you mentioned while you were describing these tests were small scale and did not were very significant, because for some tests you said that there are some clinical studies.
Yes, but in let's say small clinical studies, let's say 150 to 100 patients, maybe this assay that Alan Ramley is developing and was published in Journal of Clinical Investigation, if I remember correctly, has the greatest potential. I think they are performing some clinical studies. Maybe we can hear more about this in the coming years.
We have another question from Noemi. She says, great talk, very informative. Cholesterol efflux has been studied in macrophages, but what about vascular smooth muscle cells? That's another good point. Is there any evidence of interaction between the HDL and smooth muscle cells?
Well, mostly it is being studied with macrophages, because are cell lines, a lot of cell lines of macrophages, and can be easily used. I think there are some assays with smooth mast cells, but all these studies that examine the correlation of cardiovascular risk with cholesterol reflux capacity, they use macrophages.
Okay. Maybe some other effects are interesting when you try to incubate the smooth muscle cells, vascular smooth muscle cells, and HDL to see another type of interaction, especially because we know, at least in the past couple of years, there has been some knowledge that vascular smooth muscle cells tend to change their phenotype to something that is very close to pro-inflammatory macrophages. So this interaction would probably be interesting to study. Okay, any more questions from the audience? Nobody's raising hand. So I would like to thank you, Angeliki, so much for joining us and for giving us this nice overview of HDL and why it's important to study the function of HDL from so many different perspectives. I would just like to give an announcement that the next webinar is going to be on the 24th of March, 4 p.m. and the lecture is going to be dedicated to the ML machine learning algorithms. The title of the topic is going to be Harnessing Bioinformatics Strategies for Multiomics and Machine Learning Algorithms Integration in Cardiovascular Disease Research. Quite a title. I hope that it's going to be less complicated for us who are not ML specialists to digest the material. But in any case, join us on 24th of March, and then we can probably prepare a little bottle of aspirin below us just to combat with the headache that we will get after this lecture. I'm just kidding. Thank you so much, and see you in 1 month, more or less. Enjoy.
Thank you, Angelica.
Bye.
Thank you.
Thank you. Bye-bye.